Gregor Mendel was a monk who spent many years breeding pea plants, and his discoveries about the laws of inheritance of traits form the basis of the genetics of heredity that we still use today. Hi, and welcome to our lesson on genes and heredity. Today we're going to start with some basic terms of heredity, as well as gene information and the basics of Punnett squares. So, heredity is the passing of traits from parents to offspring. It ensures that you have similar characteristics to your parents, but not an exact copy. So, this is important because if your parents were successful enough to survive, reproduce, and pass their genes on to the next generation, their offspring should have similar traits. Genes are going to be the individual units of instruction that are located on the chromosomes that produce or influence a certain trait in the offspring. And a genome is a cell's complete endowment of DNA, so all the DNA that is in an organism's cells. So biological traits are controlled by the genes of both parents, but are unique to each individual. So there's about 8 million different combinations between the 23 chromosomes from each parent. So children are not exact copies of their parents. And every individual has alleles for their traits, which are two or more alternate forms of a gene, such as brown hair versus blonde hair, green eyes versus blue eyes, curly hair versus straight hair. And that gene's locus is its location on a specific chromosome. So when we look at these chromosomes, we can see that there's two different alleles at the second or the gene locus that's identified as C, both C and C positive. So on these two chromosomes, we have different alleles for gene A, gene B, gene C, but gene D has the same allele. So scientists did notice that traits were passed on, but nobody was able to figure out how. Aristotle suggested it was through the blood, which is why things are termed the bloodlines. Early naturalists believed that hybrids were species that were a result of breeding between other species. Buffon in the 1700s suggested that the head and limbs came from the father, while the rest of the organism came from the mother. And in the very late in the 1800s, people believed it was a blending of the traits from both parents. But in the very late 1800s, microscopes allowed for meiosis to be observed, and there was a speculation about chromosomal involvement. But before then, Gregor Mendel worked with garden peas to explain gene inheritance for plants and heredity, and he lived between 1822 and 1884. And he chose a number of visible traits that are explainable in one of two ways. So traits that only really have two expressions. Were there green or yellow peas, tall versus short plants, and wrinkled versus smooth peas? Choosing plants was pea plants was a good choice because they're both self-fertilizing and cross-pollinating. So a plant could fertilize the pistil of itself. So the male stamen attaches to the female pistil inside the same flower. Or we could have cross-fertilization. So the pollen from a male stamen attaches to the female pistil inside another flower. So cross-fertilization allows for one plant to fertilize a different plant. So we could intermingle the traits between two plants. And Mendel removed the stamen and transferred pollen from one plant to another. And he combined the male and female sex cells of different plants. So Mendel's experiments involved the crossing of pea plants with different traits to see what they would have. So we would have the initial cross, the parental cross of a round and wrinkled seed. And then the first generation would have all round seeds. But then he would cross two individuals from that F1 generation. And in the second generation, he would have 3 round seeds and 1 wrinkled seed. And that's what we're going to get into today. So Mendel wondered why the F1 generation produced all round seeds, but the F2 produced more round seeds than wrinkled seeds, but there was both there. And did it make a difference if the characteristic came from the male or female plant? In order to answer these questions, Mendel repeated the procedure with other characteristics, so those tall and short plants, green versus yellow peas, and different colored flowers as well. So Mendel discovered that one trait typically will dominate another, no matter what characteristic no matter the characteristic did it come from the male or female sex cell. So the one trait was dominant, no matter if it came from the male cell or the female cell. And he established general principles of genetics, which would later become the laws of inheritance. So Mendel's laws of inheritance included the law of unit characteristics. So traits are going to be controlled by factors called genes that occur in pairs, one from each parent. So when we have two capital R's there, we're saying that's two dominant genes. One capital R, one lowercase r would be one dominant gene and one recessive gene. And then two lowercase r's would be two recessive genes. And then there was the law of dominance. A dominant form of a trait prevents or masks the expression of the recessive form. So if you have one allele for a dominant trait and one allele for a recessive trait, you would only see the dominant trait. The dominant form is going to be represented by capital letters and the recessive form by lowercase letters. So capital R and lowercase r. Now, when you're doing your own examples, I always like to make sure I'm choosing letters that have a very different uppercase and lowercase. So choosing things like O and C can be a little bit tricky because if you're looking at somebody else's work or somebody's looking at your work, it might be difficult to distinguish between the upper and lowercase letters. The law of segregation was that during gamete formation, or the process of meiosis, the homologous pairs separate and gametes have one of each homolog. So you only have one of each allele in the gamete. And then the law of independent assortment, which is that genes are located on separate chromosomes and they separate independently during meiosis. One gene does not affect where any other gene will move. So all dominant genes do not end up in the same gamete all the time, even though it can happen. So you can have the dominant gene for one trait and the recessive gene for 6 other ones, or the dominant gene for 3 and the recessive gene for 3. It doesn't matter. So just because a trait is dominant, it does not mean that a majority of the people will express that phenotype. And the phenotype is the genetic expression. So most Dutch people happen to be blue-eyed and blonde. Those, however, are both recessive traits. So dominant genes determine the expression of the genetic trait in offspring, and recessive genes are typically overruled by dominant genes. And again, that's the lowercase letter. So normally the letters of the genes follow the dominant gene name and will stay the same for recessive genes. So let's say we have red flowers are dominant and white flowers are recessive. We would use a capital R for red and a lowercase r for white, even though white starts with a different letter. So in the last slide, we use the term phenotype. This is what's observed when we display the genes carried by an individual. So it's what we see. Does somebody have blonde hair or brown hair, blue eyes or brown eyes? And this is a result of an interaction between genes and the environment. So we can't see someone's genes, but we can see their phenotype, what they appear as. So, and genotype would be the combination of genes that are carried on the chromosome for a given trait. So what type of alleles do they have? Dominant or recessive? So the genotype on the chromosomes codes for the phenotype, what we appear. So when we have our genotype, we can be heterozygous or homozygous. Heterozygous can also be called a hybrid or a carrier. They would express the dominant phenotype. However, their genotype is one dominant allele and one recessive allele. A homozygous individual can also be called purebred. It can be homozygous dominant and have two dominant alleles, or homozygous recessive and two recessive alleles. So when we look at the offspring in phenotypes and genotypes, often we'll write them as a ratio. So typically that ratio would be homozygous dominant, heterozygous, homozygous recessive. So we might go, there might be 9 dominant, 3 heterozygotes, and 1 recessive. So we would just write that as 9, 3, 1. And the phenotypic ratio we show simply as dominant recessive. And so it would be 3 round, 1 wrinkled. So we would write that as 3 to 1. So today we're going to start with monohybrid crosses. And this is a way that we can determine the possible genotypes and phenotypes of offspring resulting in a cross. So in a Punnett square, the gametes from one parent are arranged along one side of the square, and the gametes from the other are arranged on a second side. Then, into the middle of the squares, we write the possible combinations of the gametes. Monohybrid crosses study a single trait with simple dominance over the recessive allele, and they're the easiest to study. For example, a plant that is heterozygous for seed shape is crossed with another plant that is heterozygous. So the parents would both have one dominant and one recessive allele. The possible gametes for each parent would be one dominant and one recessive. So we write for one parent, sorry. we would write with one parent along the top, and the next parent we would go down along the side of the Punnett square. When we look at the offspring, the genotypic ratio was 1 to 1. So if we go back, we have one homozygous dominant, two heterozygous, and one homozygous recessive individual. For our phenotypes, we would have the three individuals that are homozygous dominant and heterozygous that would be phenotypically dominant, and the individual that is homozygous recessive would appear with that recessive phenotypic ratio. Now that 3:1 ratio or 1 to 1 is common in every heterozygous cross. So if you get a question with two heterozygotes that are crossing, you can do the Punnett square, or you can simply remember the common ratio. So we're going to do a few practice examples, and please forgive my incredibly slow penmanship. So we are going to use the traits for seed shape round dominant to wrinkled. So R is going to be our allele of choice. So we have a homozygous dominant individual. So we're going to have two round alleles. There. And then we're going to have homozygous dominant on the bottom, or for the other parent. So we're going to have two round alleles down this side. Then we can fill in all the blocks of our Punnett square with those possible alleles. So when we look at our phenotypic and genotypic ratios, we would have 4 to 0, also known as 1 to 0 for our genotypic ratio. And for our phenotypic ratio, we would also have 4 to 0 or 1 to 0 for our phenotypic ratio for that first cross. Okay, I'm going to erase these ones so that I have room to do my next Punnett square. Okay, so for our next cross, we have homozygous dominant. So again, two round dominant round alleles, and then we have a heterozygous plant that we're going next. So we would have homozygous dominant, another homozygous dominant, and then two heterozygous offspring there. So when we're looking at our second example, we would have a phenotypic ratio of 2 to 2 to 0, which we can then write again as 1 to 1 to 0, just because then it's in the lowest common terms. And then our phenotypic ratio would still be 4 to 0. And so we would write that again as 1 to 0. So it's in our lowest common terms. Okay, so we have three different Punnett squares to do here, a homozygous dominant and homozygous recessive. So we would have our two dominant alleles here. You can put them down the side. I just always start with writing on the top. And then homozygous recessive. So when we fill in our Punnett square, we would have 4 heterozygous. That's supposed to be an R. I said 4. So we would have a ratio of 0, 1, 0 for genotype, and then 1 to 0 for phenotype. On our next one, our heterozygous versus homozygous recessive. Pen. We would have our heterozygous versus our homozygous recessive. And we would fill everything in. So we combine all the squares. So we would have a genotypic ratio of 0 to 1 to 1. Again, our lowest common denominators, and a phenotypic ratio of 1 to 1. Okay, and now our last one, homozygous recessive, and I almost am running out of room. And homozygous recessive, and everything we fill in is going to be recessive. So we would have a genotypic of 0, 0, 1, and a phenotypic ratio of 0 to 1. So everything's in its lowest common denominator. Okay, so now we have one that we're going to have to do different letters for. So we're going to determine the phenotypic and genotypic ratios between a red flower and a recessive white flower. So again, I'm going to have my large R for red and my lowercase r for white. And it's important we keep the same letter because later on we're going to talk about ones that are codominant, so then we need different letters. So we have a homozygous dominant and a homozygous recessive white flower. So all of our offspring are going to be heterozygous. So they're all going to have we're going to have a ratio of 0, 1, 0 for our genotype, and 1, 0 for our phenotype, and all of our flowers are going to be red. Okay, last one here is the phenotypic and genotypic ratios between a man who is heterozygous for skin pigment and a woman who is albino for skin pigment. So I'm going to use S for pigment and lowercase s for pigment. And again, I said not to use ones that are too obvious, but S and P both have similars, so I'm just going to stick with S today. So he's heterozygous, so he has one capital S, one lowercase s, and she has no skin pigment, so she's recessive. So she's two lowercase. So we have big S, little s, big S, little s. So I'm making it super obvious. Big S, little s. Sorry, this was supposed to be little s, so I need to erase that one. Little s, little s. So our genotypic ratio is going to be 0, 1, 1, and our phenotypic ratio is going to be 1 to 1. So 2 of the offspring are going to have skin pigment, 2 of the offspring will be albino. Okay, a test cross is performed to determine the genotype of a dominant phenotype by looking at them, we can't tell if they're homozygous or heterozygous. So a test cross is performed to determine the unknown genotype. So we'll cross a dominant individual with a homozygous recessive individual. If the unknown is heterozygous, half of the offspring will be dominant and half will be recessive. Now, this doesn't work out perfectly, but Mendel had a lot of time to crossbreed a lot of peas. And if the unknown dominant trait is homozygous dominant, all of the offspring will be dominant. And we'll see that in these 2 test crosses. So a farmer wants to know if his white sheep is purebred or a hybrid, so he crosses it with a black sheep. So the white sheep has a uppercase W for white and an unknown second allele. Where the black sheep will have 2 lowercase w's because they're unknown. We can see in this first test cross where the white sheep was heterozygous, we're going to have 2 offspring with the dominant trait and 2 offspring with the recessive black trait. Now, where that unknown test cross was homozygous dominant, we have all 4 of our individuals are heterozygous, so they will all have the dominant trait. So that was our introduction to Mendel's genetics and Punnett squares. I hope you enjoyed it.